Welcome back to the Deep Dive. Today we are tackling something, well, pretty surprising in cancer biology.
It really shifts the perspective, doesn't it?
It does. For, decades, we've known that cancer cells often show up near nerves.
Mm-hmm.
The tumor gets innervated.
Mm-hmm. And we also knew, clinically, that having lots of nerves in the tumor, that usually meant worse outcomes for patients, more metastasis especially.
The link was definitely there.
Mm.
But how it worked, that was the mystery, wasn't it?
Exactly. Was it just bad luck the nerves were nearby, or was something active happening?
And then studies started showing if you targeted those nerves, denervation they call it.
That's the term, denervation. If you did that, you could actually slow down the tumor's growth. Okay, so that's a big clue. But it still leaves the question. What's the actual conversation? What are the nerves and cancer cells, exchanging?
Right.
If there's this critical communication happening right there at the nerve-cancer interface, what's being passed back and forth?
What is the nervous system handing over that helps the tumor not just grow but spread?
Well, the answer that came out of the research we're looking at today, it's pretty stunning. The nervous system isn't just like passive scaffolding. It's an active supplier. It's literally feeding the tumor the cell's power plants.
Wait, power plants, you mean?
Mitochondria, the actual mitochondria.
Wow, okay. So that's huge. The nervous system is basically giving an energy bailout to the cancer. That's a great way to put it. An energy bailout. It's the key insight here. This non-cell autonomous metabolic plasticity. Non-cell autonomous, meaning the cancer cell isn't just managing its own energy.
Exactly. It's not just rewiring things internally. The nervous system is like outsourcing the energy supply. It's physically handing over the fuel needed for metastasis.
Which is the most dangerous part of cancer, really.
Absolutely. That journey is incredibly demanding. So our mission today is to unpack how this works, the evidence, the actual mechanism of transfer, and crucially, what this energy boost does for the cancer cell. All right, let's dive in. Section 1. Cancer needing nerves and this idea of metabolic plasticity. Okay, so to really get why transferring whole mitochondria is such a big deal, we need to talk about metabolic plasticity.
Right. Cancer cells, they're famous for the Warburg effect, aren't they? That fast, kind of inefficient energy production, glycolysis.
Yeah, they burn sugar really fast, but don't get a lot of bang for their buck, energetically speaking. That works okay in the main tumor, maybe.
But metastasis.
Yeah.
That's a whole different ballgame. It's brutal out there for a cancer cell.
Totally brutal. When a cell breaks off, floats through the blood, tries to set up shop somewhere new, it needs serious, sustained, efficient energy.
So it can't just rely on that fast-burn glycolysis anymore.
Not really. To survive all that stress, lack of nutrients, oxidative stress, physical forces, it needs to switch gears. It needs to rely heavily on oxidative phosphorylation, or OXPHOS.
And OX6THOS. That happens inside the mitochondria. That's the efficient, high-yield energy pathway.
Exactly. So this ability to switch fuel sources to adapt its metabolism based on the situation, that's metabolic plasticity, and it's absolutely critical for a cancer cell to successfully metastasize.
Okay, so the cell needs this high-powered OXPHOS capacity. Where does it get? This brings us back to the nerves, right? The evidence for dependency.
Yeah, the evidence is really solid. Our sources point to studies in human prostate cancer, aggressive mouse breast cancer models.
And in those studies, when they removed the nerve supply.
Targeted nerve withdrawal, yeah. It clearly hampered tumor growth.
And how did they do that? How did they remove the nerves? They used a specific chemical tool, botulinum neurotoxin type A, Bonte. Most people know that as Botox for wrinkles.
Right, but here it's used scientifically. Bonte works by stopping nerves from releasing their chemical signals, their neurotransmitters.
So it basically silences the nerve communication.
It effectively silences them. And when the researchers did this, the cancers showed immediate signs of metabolic trouble.
Metabolic trouble how?
Well, they looked at the genes being expressed, and there was this major downregulation of mitochondrial metabolism. The cancer cells were sort of forced back onto that less efficient glycolysis pathway.
They lost their high-efficiency engine.
Pretty much, and they measured it very precisely. In one model, a type of breast cancer called ductal carcinoma in situ,
Okay.
denervation hit the tricarboxylic acid cycle, the TCA cycle, the hardest. That's like the absolute core of mitochondrial energy production.
And did that have a real-world effect on the tumor?
Oh, absolutely. A dramatic effect. In the mice where they denervated the tumors, the number of invasive lesions, the parts of the cancer actually breaking out, dropped from about 55%.
Wow.
Down to just 12%.
So that nerve-supplied metabolic boost wasn't just helpful, it was necessary for the cancer to even become properly invasive.
It really seems that way.
Yeah.
Critical for that invasive potential.
Okay, let's just pause for a sec. Quick recap. We know cancer needs efficient energy, OXPHOS, from mitochondria, especially to invade and spread.
Right.
And we know that nerves somehow provide critical support for that. Taking away the nerves messes up the cancer's mitochondrial metabolism.
Mm-hmm. Which leads us right to the next big challenge. If nerves are supplying this energy boost, how do we prove they're physically handing over mitochondria, these tiny organelles?
Yeah, how do you track that in real time?
Yeah.
And maybe even more importantly, how do you show that a mitochondrion that's been passed over still works in its new home?
That's the million-dollar question they tackled next. All right, section 2. The actual transfer mechanism and proving this metabolic boost. How did they figure this out? They set up a really neat system in the lab. They co-cultured aggressive breast cancer cells, the 4T1 line, with neuronal stem cells.
Stem cells that can become neurons.
Exactly. They nudged these stem cells to differentiate into mature neurons right there alongside the cancer cells.
And what happened when they were together?
Well, here's where it gets really interesting. It wasn't just the cancer cells changing. The neurons responded dramatically.
How so?
It seems like the cancer cells put out this huge demand signal, like, we need energy. And this pushed the neuronal precursor cells to mature and differentiate faster. And to do that, the neurons themselves had to massively ramp up their own energy production.
They had to build up a stockpile.
That's a good way to think about it. They measured the amount of mitochondrial DNA, mtDNA, in the neurons. Before interacting with cancer cells, it was around 16 copies per neuron.
Okay, a baseline.
After being co-cultured and induced to differentiate by the cancer cells, that number jumped to, get this, 226 copies per neuron.
Whoa. That's a huge increase. Like the neuron went into overdrive.
Serious overdrive.
Yeah?
Becoming a hyperproducer of mitochondria.
Did the mitochondria look different too?
They did. Visually, they changed shape. In the undifferentiated cells, they were kind of round, globular, but when the cancer cells were around, they stretched out, becoming thin, elongated tubular structures.
And that shape means?
That's a classic sign of mitochondria that are really geared up for high metabolic output. For OXPHOS, they're optimizing for energy production.
Okay, so the neuron is loaded, primed with these high-energy mitochondria. How does the handoff happen? Is it just chemicals floating across?
No, it seems to be primarily physical contact. Direct cell-to-cell connection.
Like holding hands.
Sort of. Often through these incredibly fine temporary bridges called tunneling nanotube-like structures, or TNTS.
Wow, actual tubes between the cells.
Yeah, microscopic tunnels acting as direct conduits. They tested this too. They used special culture plates with a barrier called transwell inserts that kept the cells physically separated but allowed chemicals to pass.
Right.
When direct contact was blocked, the mitochondrial transfer basically stopped. So physical connection seems key.
The physical evidence looks strong. But like you said, you need to prove they work. You can pass over a battery, but if it's dead,
Yeah, exactly.
the functionality is everything. And this leads to, I think, one of the most elegant experiments in the study, the row zero cell experiment. Row zero. Okay, what does that mean?
Row zero cells are engineered to be metabolic weaklings. They took those 4T1 cancer cells and genetically stripped out all their mitochondrial DNA. Completely gone.
So no mtDNA means?
No OXPHOS. They completely lose the ability to do efficient respiration. Their mitochondria look fragmented, globular again.
And they can't survive easily.
Right. They become dependent on supplements. Specifically, they need extra uridine added to their culture medium just to stay alive and multiply. They're metabolically crippled.
Okay, the ultimate test subject. What happened when they put these crippled caudal cells together with the healthy neurons?
It was a rescue mission. The neurons started transferring their healthy mitochondria over to the caudal cancer cells.
You could see it happening.
Yes. Over time, the 9-read dollar cells started reacquiring mitochondrial DNA. Their mitochondria started looking normal, again, elongated and healthy.
But the real test, did they regain function? Could they live without the supplement?
That was the clincher. They took away the uridine life support, and the rescued $2 cells. They didn't just survive, they started forming colonies, growing, and they regained their full OXPHOS capacity.
Wow.
It proves that the mitochondria transferred from the neurons weren't just present, they were fully functional. They integrated into the cancer cell and restored its energy production. They were breathing again, metabolically speaking.
That's incredibly convincing for the lab setting. But how do you track this in a whole animal or even look for it in human patients? Regular fluorescent tags just fade out, right?
They do. The signal gets diluted with each cell division. If you want to know the ultimate fate of the cells that received mitochondria, you need something permanent, a way to tattoo those cells and all their descendants.
Like a lineage marker.
Exactly. And that's why they developed this ingenious tool called MitoTRACER.
MitoTRACER. Okay, sounds cool. How does it work?
It's basically a genetic switch that gets flipped permanently when a mitochondrion is transferred. It's quite clever.
Walk us through it. This sounds important.
Okay, imagine two parts. The donor neuron is engineered so its mitochondria have a special protein attached to them, Cre recombinase. Think of Cre as a molecular scissor.
Okay, scissors on the mitochondria.
Now, the recipient cancer cell is set up like a traffic light. It's normally making a red fluorescent protein, so it glows red. But the gene for red is followed immediately by a SACTP signal. And after the STP signal is the gene for a green fluorescent protein.
So it's stuck on red because of the STP sign.
Right. The cancer cell also makes another protein, a key, called TEV protease. TEVP.
A key for the scissor.
Sort of. When a neuron's mitochondrion, with the Cre scissor, enters the cancer cell, the TEDP key finds the Cre and cuts it loose from the mitochondrion.
Okay, the scissor is free.
Now, the free Cre scissor can travel into the cancer cell's nucleus, and there it finds that red stop green gene sequence.
And it cuts.
It cuts out the S.OP signal, permanently removes it from the DNA.
So the cell stops making red protein.
And starts making green protein instead. It switches irreversibly from red to green.
And because it's a change to the DNA.
Every single cell that descends from that original recipient cell will also be green. It's a permanent lineage trace. You know that any green cell you find came from an ancestor that received a mitochondrion from a neuron.
That's brilliant. A permanent marker for the transfer event. Did they see this working beyond the lab dish? Like in people?
Well, they looked at human prostate cancer samples using advanced imaging, and they found that cancer cells physically closer to nerves. They had significantly more mitochondria than cells further away.
Consistent with the idea of local transfer.
Exactly. And when they treated similar samples with Bonti, the nerve-silencing toxin, that higher mitochondrial load near the nerves, went down.
Connecting the nerves to the mitochondria levels.
And then in mouse models, they used sophisticated sequencing, nanopore sequencing, to actually detect mouse mitochondrial DNA inside the human cancer cells that had been implanted.
Direct proof of host-to-tumor transfer.
Direct proof of transfer in vivo. And, again, hitting the nerves with Bontinet reduced that transfer by about 35%. It's happening in living systems.
Okay, another quick pause. This is getting complex, but really fascinating. We've got proof of transfer now, often via these tunneling nanotubes.
Yeah.
We know the transfer mitochondria are functional. They can rescue metabolically crippled cells.
Check.
And we have this MitoTRACER system to permanently label and track the cells that receive the mitochondrial gift, even in a living animal.
Check. All pieces in place.
So the big question that MitoTRACER lets us finally ask is, what's the payoff? What actual advantage does getting these neuronal mitochondria give a cancer cell, especially when it's trying to metastasize? What does this gift let it do?
That's exactly where the research went next. What's the functional consequence? All right, section 4, the advantage and the fate mapping. They used MitoTRACER and cell sorting to isolate the green cells the recipients write. Yeah, they used FACS sorting to physically separate the green cells, the ones that got mitochondria, from the red cells, the ones that didn't, from the same tumor. Then they could compare them head to head.
And what were the differences? Did the green cells have superpowers?
You could almost say that. They definitely showed characteristics linked to more aggressive cancer. For one, they were better at growing without sticking to a surface anchorage-independent growth.
Which is important for cells floating in the bloodstream.
Exactly. And they were much better at forming mammospheres in culture.
Mammospheres. That's a marker for cancer stem cells, isn't it?
It is. It suggests these recipient cells gain more stem-like properties, more self-renewal potential. Metabolically, they were just supercharged.
How so?
Enhanced OXPHOS capacity, better coupling between oxygen consumption and ATP production, just higher overall energy levels. They were running on premium fuel.
But metastasis isn't just about having energy. It's about surviving really harsh conditions. Did they test resistance?
They did, and this is maybe the most crucial finding for metastasis. The green cells had a much better redox balance.
Redox balance,
Yeah.
meaning how the cell handles damaging molecules.
Exactly. Specifically, they looked at glutathione, the cell's main antioxidant. The green cells had a higher ratio of the protective reduced form, GSH, compared to the used oxidized form, GSSG.
So they had a stronger shield against damage.
A much stronger shield. And this translated directly into better survival against two major stresses that metastatic cells face.
Which are?
First, oxidative stress. They treated the cells with hydrogen peroxide, which mimics the damaging environment when cells detach. The green cells handled it much better.
Okay, and the second?
Shear stress. This mimics the physical battering cells endure being squeezed through tiny capillaries and whipped around in the bloodstream. Again, the green cells were significantly more resistant.
So the mitochondria didn't just give them energy, they gave them armor.
Energy and armor. A resilience package deal, basically.
Wow. Okay, so the final piece of the puzzle. Tracking these cells in the body. Did the green cells actually succeed more often in metastasizing? This is the fate mapping part, using MitoTRACER in vivo. This is where it all comes together. They let the tumors grow in mice, waited for metastases to form, and then looked at the proportion of green cells.
What did they find?
The numbers are really stark. In the main primary tumor, only about 5.4% of the cancer cells were at the back. Green had received mitochondria, a relatively small fraction.
Okay, a minority population. What about in the metastases?
That's where the selection becomes obvious. In metastases that formed in the lung, the proportion of green cells jumped to 27.3%.
Wow, like a five-fold enrichment.
And even more dramatically, in metastases that formed in the brain, it was 46.0%. Nearly half the cells in the brain metastases originated from cells that had received mitochondria from neurons back in the primary tumor.
That's incredible. It's powerful proof that getting those mitochondria provides a massive survival and colonization advantage during metastasis. Absolutely. It strongly suggests that this transfer is a key event that selects for the cells most capable of completing the metastatic journey. And they saw similar enrichment in a melanoma model too, suggesting it's not just unique to breast cancer.
So, wrapping this up, the big picture conclusion.
I mean, the synthesis here really changes how we should think about the tumor microenvironment and the nervous system's role. It's not just adjacent, it's actively participating.
Providing an adaptive arsenal, as the researchers put it.
That's the perfect phrase. This mitochondrial transfer equips the cancer cells with higher metabolic efficiency and better stress resilience. Both are absolutely essential to survive that incredibly inefficient, deadly process of metastasis. The nervous system is, maybe unintentionally, a critical partner in crime.
And that huge enrichment in the brain, nearly 50%, that seems particularly telling.
Neurons are metabolic powerhouses themselves, right? Exactly. They're optimized for efficient energy use. So if a cancer cell gets mitochondria from a neuron, it might be getting a particularly high-quality, high-performance energy source compared to, say, mitochondria from other cell types. It's like getting a Formula One engine.
Especially helpful for colonizing the energy-hungry brain environment.
Plausibly, yes.
So this leaves us with a really provocative final thought, doesn't it? If nerves are acting like this,
Yeah.
like a metabolic sugar daddy, as the outline called it,
Yes.
and this physical transfer through things like nanotubes is key for selecting the most dangerous metastatic cells. What does that mean for treatment?
Well, that's the next frontier, isn't it? If we can understand exactly how these TNTS, these tunneling nanotubes, form between nerves and cancer cells.
Could we block them?
Could we develop drugs that specifically prevent those physical connections from forming? Snip the tubes, maybe. Could we cut off that energetic lifeline and essentially starve the most aggressive metastasis-initiating cancer cells of the very power they need to spread?
Wow. Blocking the energy transfer itself. That's a fascinating angle for future therapies.
It really opens up new possibilities.